All right, welcome everybody. We'll still kind of admit people as they trickle in, but I want to thank you all so much for being here tonight. We are very excited to learn about stormwater and cyanobacteria as well. I hope you guys are too. I'm Hannah Mahar. I'm the current FNLC or Friends of Northern Lake Champlain Eco AmeriCorps member. And my co-host for tonight is Sarah Gratz. She's the Eco AmeriCorps member over at Northwest Regional Planning Commission. So a few kind of housekeeping items before we begin. The webinar is currently being recorded and it's going to be posted on the Franklin Stormwater Collaborative website. There will be a link for the website placed into the chat, so feel free to go check that out after the webinar is completed. As far as questions go, you'll see a little kind of Q&A button at the bottom of the screen, and if you have any questions that pop up during the webinar, feel free to just throw them into the Q&A and we'll get to them at the end of the presentation. Also, if you'd like to ask something in person, once the presentation is over, you can unmute yourself and also ask a question that way. And last but not least, just for the duration of the presentation, please keep yourself on mute. But other than that, I'm going to hand it over to Peter and stop sharing my screen.
All right, thank you, Hannah. And I see a few people drifting in, which is great. It's really nice to talk about this stuff. It's a good time of year to talk about it because it feels like a long way away, but very soon we're going to have to start signing up new volunteer cyanobacteria monitors and getting the new season going. So it's nice to get a jump start on that now. So I will share my screen here if it works. Sorry, I have to give permissions, I guess.
You should have permission.
I think my computer needed a permission.
Oh, okay.
All right, sure, let's try that again. There we go. All right, can you see my presentation now?
Yes.
It's the beginning. So yeah, Sarah asked me if I would give a presentation to this group to talk about cyanobacteria blooms, particularly as they relate to some of the work that she's doing and maybe some of you are doing with sort of stormwater stuff, and kind of more generally, I think, to give an idea of sort of the end problem that's being addressed with a lot of this kind of on the ground stormwater type work. And as I'm sure many of you are aware, a lot of that work in Vermont and in the Champlain Basin is really targeted at reducing the incidence of cyanobacteria blooms in the lake, and of course in other lakes around Vermont and around the world. This is an important issue. So for this talk, I'll give you a brief outline. First, I'll just give a little background on the science of cyanobacteria blooms, sort of what causes them, a little bit about the basic ecology of cyanobacteria, and sort of what their distribution is like around the world, and more particularly in Vermont, using some of our monitoring data, largely collected by our volunteer cyanobacteria monitors. Then I'll try to touch on some issues related to stormwater and their impacts on lakes. I don't know, maybe some of this will be old hat to some of you. This is not really my field of expertise, but of course, stormwater is one of the major sources of nutrients to the lake. And when it comes to cyanobacteria blooms, it's really largely about nutrients, although things like climate also play a big role. And then I'm going to touch a little bit on emerging topics related to cyanobacteria in Vermont, sort of ongoing research efforts that I think are exciting, and just things that we're trying to do in the monitoring program to sort of improve our understanding of cyanobacteria blooms in the state. Yeah, mostly just stuff that I'm excited about that I thought I'd share. A little bit about myself. Who am I? So I work for the Vermont DEC, the Department of Environmental Conservation, in the Watershed Management Division, particularly in the Lakes and Ponds program. And there I'm the project lead on the Lake Champlain long-term monitoring project, as well as the cyanobacteria monitoring project, which covers Lake Champlain as well as inland Vermont lakes. So those are kind of my main hats that I wear in the department, and then sort of it being a small state and a pretty small lakes program, I'm inevitably weigh in on a number of other kind of lakes related things around the state. Before working here, where I've been for coming up on 3 years now, I did my PhD at UVM studying cyanobacteria blooms, really focused on Missisquoi Bay and the rest of the lake as well, but largely Missisquoi Bay. Then I did a couple of postdocs in Sweden and Switzerland where I studied a wide kind of range of lake-related issues related to lake biogeochemistry, cyanobacteria bloom ecology, changing climate, developing new monitoring tools and things like that. So really a research background. So that sort of research focus and monitoring focus is kind of my real expertise, but of course, inevitably now I'm getting a little bit more into the management side of things. All right, so jumping into sort of the basic ecology of cyanobacteria, I think many of you are probably aware that cyanobacteria are a unicellular organism, so they're very simple-celled organisms. They're often colonial, and they're capable of photosynthesis, so they're distinguished from other types of bacteria by that ability to photosynthesize. I always say, cyanobacteria have been around for billions of years. They're responsible for the oxygen in our atmosphere, and they live pretty much everywhere on Earth. So in and of themselves, they're a natural and important part of ecosystems. It's really always, as with so many things, a question of balance. And when we get runaway growth of cyanobacteria, then we can start to run into problems. So in Vermont, we have a number of sort of common cyanobacteria species that form cyanobacteria blooms. These are really cosmopolitan species, so they tend to occur around the world, and they, you can find papers on microcystis blooms on every continent. The same for Dolichospermum, Phanosomenon. Boeotrichia doesn't typically form huge blooms, but occasionally it does. So these are all colonial forms of cyanobacteria. This is a common feature of most of the problem types of cyanobacteria. So they can form large colonies, and importantly, they can also regulate their buoyancy. So this means that all of these types of cyanobacteria have within their cells these essentially little air bubbles called gas vesicles, which they can produce and which allow them to float up to the surface under calm conditions. And so this is an adaptation that other types of phytoplankton or aquatic algae do not have. So this gives them a competitive advantage over other groups under certain circumstances, particularly during calm conditions when that ability to float up to the surface is strong enough to overcome turbulence. If it's a windy day, of course, that ability to float isn't strong enough, and they'll still be mixed, just like any other plankton would be. So these are just a taste of the species. There's many more, but I would say that sort of these groups here and maybe 2 or 3 more account for 95% of the biomass of cyanobacteria that we see in Vermont lakes. So those are some of the cyanobacteria that we see, but what are cyanobacteria blooms? So blooms are a poorly defined term. I mean, I think that somebody who's used to living on Saint Albans Bay might identify a bloom differently than somebody who's living on a sort of more pristine, clear water, smaller lake in Vermont. So, this is a problem. This sort of, you should call it dependence on your sort of frame of reference is a sort of a problem in the literature. So there's not a clear definition on what constitutes a bloom from a kind of ecological perspective, although from a health perspective, there are thresholds associated with cell density and toxin concentrations that are used to define blooms sort of at the level of the World Health Organization and other institutions like that. In order to get a cyanobacteria blooms, first, you need to have enough nutrients and light to let the bloom grow. So you need the basic kind of elements of that recipe. But then you also need the right weather conditions to allow those populations of cells to concentrate. So it's both a question of how much, how many cells are in the water column, but then you also need the right hydrodynamic conditions to allow them to accumulate in one place to get the really high cell densities that are really associated with human health impacts. So typically, blooms are an issue only when there are excess nutrients in lakes from human activities. Maybe I shouldn't say only, but for the most part. And the nutrients that fertilize blooms are the same basic nutrients that fertilize, farm fields and terrestrial plants. You have nitrogen and phosphorus. Those are the sort of two main nutrients. So in contrast to terrestrial systems where nitrogen is most commonly the limiting resource, in lakes and other freshwater systems, phosphorus is typically the limiting resource. So if you increase phosphorus, you can typically increase the biomass of plankton and cyanobacteria. However, a recent thing that has been occurring in many parts of the country is more reports of blooms from oligotrophic lakes. Oligotrophic means nutrient poor lakes. So we've seen this sort of locally in the Adirondacks and some lakes that have no development in their catchments. More cyanobacteria blooms have been observed. And we may see hints of this in Vermont as well, in some of our more remote lakes where we don't have a lot of things like agriculture in the catchments. We're still seeing some blooms take place. So this is something that is interesting to look into, and there's a strong suggestion that climate probably plays a role there, but there may be other factors like atmospheric deposition of different nutrients as well. So just to kind of go back again to what causes cyanobacteria blooms, I always sort of come back to the analogy of baking cookies. So if you have a recipe for cookies, you might have your basic ingredients like flour, sugar, eggs, nuts, butter, or chocolate chips instead of nuts maybe, depending on your preference. Then you'll cook them for a certain amount of time at a certain temperature to get your cookies. So in this analogy, you can think of the basic ingredients for cyanobacteria or other plankton biomass being these sort of key elements: carbon, nitrogen, and phosphorus. And that is sunlight as well. Sunlight is linked to carbon through photosynthesis. And then you have a range of other micronutrients that typically are more important for driving blooms in kind of large nutrient poor systems. But in a system like Champlain, those are typically abundant enough that they're not going to limit production. And as I said before, in lake systems, and particularly in Lake Champlain, it seems that phosphorus is most frequently limiting production of cyanobacteria, although nitrogen occasionally is, particularly during the summer. During blooms, nitrogen can become limiting nutrient pretty quickly. And as with baking cookies, you need to have these ingredients present at the right temperature conditions for the right amount of time in order to result in a bloom. So there's this real temporal aspect and climate aspect built into that recipe. And then, as I said, you have to have the right conditions result to allow those cells to accumulate in a single place. So what typically happens is you'll have a lot of growth of cyanobacteria early in the season when there's a lot of nutrients present. Those are typically mixed throughout the water column, by wind and wave activity, so that they might not be very observable until you get a sunny day when they can all rise to the surface and a bloom will seemingly come out of nowhere. So in many parts of the lake, particularly sort of the main lake basin, you'll get these ephemeral blooms that result from these accumulations of cyanobacteria that have been spread throughout the lake, but then are concentrated and wash up on shore. So the evidence shows that cyanobacteria blooms are becoming more common worldwide. There's pretty good consensus on this issue. It's not the easiest thing to study because, of course, most of the data that we have comes from lakes where blooms are a problem. So there's a certain amount of bias in the published literature. But if you look at things like sediment cores, which is what this study used, and look at the pigments of cyanobacteria preserved in those sediment cores, you can see an increase over time, over the last couple hundred years. Similarly, if you look at the satellite record, you'll see in many places increasing cyanobacteria in kind of the last 30 years or so. So here in this figure, anywhere you see a red symbol, that's an increasing trend. Although you will see in places like Europe and North America, there are more decreasing trends present. This is associated with sort of the implementation of the Clean Water Act and other similar measures in other in Europe and other parts of the world to reduce nutrient pollution as we got a better understanding of what drive cyanobacteria growth. So typically speaking, we're seeing an increase in blooms, although this is not universal. And there's pretty much a consensus that nutrient inputs are the primary driver of blooms, but that climate change is an additional stressor which will require stricter nutrient controls from a management perspective. Essentially, if we want to get the same outcome in a climate change context, we have to have more aggressive nutrient reduction targets to reach our water quality goals. I also just want to point out that this is not just a lakes problem. We are also seeing a major increase in phytoplankton blooms in the world's oceans. Pretty much all the coastal oceans have seen signs of this, particularly in the 21st century. And this is strongly linked to climate change, but of course, nutrient pollution from the adjacent terrestrial systems also plays a big role here. So how do these trends play out in Vermont? So here it gets a little tricky to look at given the data that we have. So what you're looking at here is data from the Vermont Cyanobacteria Monitoring Program. So these are reports collected by volunteer monitors from around the state that report either generally safe conditions, and that's represented by the green, low alert blooms. So these are conditions when you see some green accumulation around the lake shorelines typically, but not enough to be what we would consider a severe bloom. And then red are those high alert blooms when we would strongly recommend really staying away from the water for all activities if possible. So you see this is a time series from 2013 to 2022 for each of the sort of main sections of Lake Champlain that we monitor. And what you can see is that the percentage of bloom conditions does not show a clear trend towards increasing frequency in any of those lake basins. So there's definitely differences between different parts of the lake. As I'm sure many of you are aware, you see more blooms in places like Missisquoi Bay and Saint Albans Bay than you do in other parts of the lake. But there's not a clear indication that those are getting more frequent over time. The same is true for the inland lakes where we have enough data. We don't have very much data from inland lakes. This monitoring program started on Lake Champlain, has only kind of recently been expanded to smaller lakes in Vermont. So what we see is, there may be some indications in places like Memphremagog of slightly more frequent blooms, but this could partially reflect kind of a better understanding on the part of our monitors of where to look for blooms. So there's a certain amount of, it's hard to have perfect data in this respect. Just to look at how our inland lake data collection goes. This is sort of our recent progress in terms of trying to increase the reporting on inland lakes in Vermont. We'd like to continue this trend. So as you can see, up until about 2015, we had very few reports, just from a handful of lakes where there were certain problem conditions that we wanted to keep an eye on. But my predecessor did a great job of bringing on board more volunteer monitors, and we're trying to continue that effort, particularly through partnerships with the state parks and sort of other lake monitoring programs. So my goal would be to have an idea of bloom occurrence on as many lakes in the state as possible. Even if we aren't seeing blooms, it's important to know that we aren't seeing them, so that when the blooms start to appear, we'll know that this is an indication that things have actually gotten worse, which is very hard to say if we don't have any data from those lakes. So this is just a snapshot of the lakes. I don't need to discuss this. I think this is a holdover from a previous presentation. So as always, we are interested in getting more volunteer monitors. If you are interested, please get in touch. Most of our monitors on Lake Champlain are coordinated through the Lake Champlain Committee, which is a nonprofit that does a great job in training and keeping in touch with volunteers, doing work, doing the monitoring on Lake Champlain and making sure that, people are not duplicating sites and trying to get all the important parts of the lake covered. And if you're interested in monitoring other Vermont lakes, you can get in touch with me at this website here. So that was that should give a sense of kind of where we are in terms of bloom frequency in Vermont, although I'm very happy to discuss it in a lot more detail in the question and answer period. I think it's nice that we have a small enough group that I'm very happy to get into some discussion if there's time. But I want to touch on the role of stormwater in this particularly because in my understanding, that's really what a number of members of this group or this audience are interested in. So in terms of why stormwater is important for Lake Champlain, there's a figure from the 2021 State of the Lake report that the Basin Program puts out. And what you can see here is that in terms of the annual load of phosphorus to the lake, this is the circle in the upper left, we have 16% of that is exported to the lake from developed land. So this is, urban areas, impervious surfaces, things like that. So it's a reasonable piece of the pie. But you see that developed land is also in the next circle is only making up 4% of the total land cover of the basin. So 4% of the land cover is contributing 16% of the total load of nutrients. And if you look at the bar plot there on the right, you see that the amount of phosphorus delivered per square mile or per acre is almost twice as high for developed land as it is for agricultural land. So we frequently think of phosphorus pollution as an agricultural problem, and it is because we have a lot more agricultural than developed land in the basin. Agriculture is a very important piece of it, but developed lands are also an important piece of it. And to the extent that we see increased development, that's likely going to have some major impacts for water quality in the lake. Another important thing to note, which I don't dig into in the Basin Program report, is that stormwater tends to have high phosphorus but low nitrogen relative to agricultural land, which frequently has very high nitrogen inputs. When you have a low ratio of nitrogen to phosphorus, as you do in stormwater, this is also another factor that can predispose lakes to cyanobacteria blooms. So in situations where lakes are nitrogen poor, cyanobacteria have a competitive advantage over other types of phytoplankton because they can fix atmospheric nitrogen through specialized cells, and there are no other phytoplankton groups capable of doing that. So in a situation where we have loading from these developed lands with low nitrogen to phosphorus ratios, this is kind of compounding the problem of phosphorus pollution and predisposing the lakes even more to cyanobacteria blooms. In terms of how stormwater management practices can help, it's very hard to generalize because there's a lot of site-specific factors that go into which stormwater practices are going to be the most important. It's going to depend a lot on the percentage of impervious surface, the slope, the soil type, and other factors. Really, a lot of these practices are trying to get the surface water to infiltrate into the soil. Once it does that, then the nutrients carried in that surface water typically bind pretty strongly to sediment particles, particularly fine iron and manganese incorporated in clay minerals and other things. And once it binds to these clay particles, it's typically hard to release, except for under certain conditions. So if you can get that water to go into the land before running out into a stream, then it's probably going to come out of into the stream a lot cleaner than if it flows over the surface of the land. So yeah, that's the goal of a lot of the stormwater management practices, particularly the ones that can be done at the scale of an individual landowner or business owner. There are a lot of different levels at which these remediation actions can happen. So things like rain gardens, infiltration wells, rain barrels, keeping your lawns natural, avoiding lawn fertilizers, allowing your lawns to grow taller. All these things can improve infiltration, erosion control as well. And it's important to infiltrate as much as you can so that you have less runoff happening during storm events. What we see when we're looking at loading in streams to the lake is that the concentration of phosphorus increases with the level of discharge. So this is kind of like a double whammy. So not only do you have more water coming in, but the water that's coming in is more polluted. So it's kind of a compounding effect. So this means that if you can slow that water's progress over to landscape so that the peaks in those streams are lower, you're also going to get lower concentrations of water, so you get much lower loading to the lake. This is a particular problem in areas where you have things like combined sewer overflows, where there, of course, if you have a lower volume of water going into those sewer overflows, you can reduce the chances that that it will overflow and carry sewage out into the lake, which of course is a whole other set of problems. There are also some community level actions, things like treatment of stormwater ponds. These are kind of active areas of research. The Basin Program has funded some of this work, looking at trying to line stormwater ponds or their outflows with different minerals or types of crushed stone, which will absorb the phosphorus before it flows out into the stream network. But of course, those things are frequently pretty expensive. And then there's some sort of municipal level actions. One of the ones that I think is really interesting is actually street sweeping practices. There's some really interesting research that come out recently from the long-term ecological research network station in Minneapolis and St. Paul. So this is an urban ecological research site. And they've been looking into different leaf removal practices and how that affects total nutrient loading from stormwater systems in Minneapolis and St. Paul. And what they see is that, by using the appropriate leaf removal and street sweeping practices, they can reduce annual phosphorus loading by 60 or 70%. And this, of course, is really dependent on what time of year you're doing these actions. So particularly in early summer, when you get a lot of those, flowers that are released from the trees onto the landscape, those can get swept into storm drains. And then, of course, in the fall, when the trees drop a lot of their leaves. Yes, so I don't always think of sort of this leaf pollution as being a major source of nutrients, but actually when it comes to the stormwater nutrient load, it is pretty important. As these things are swept into storm drains and they decompose, they release nutrients, and then those are flushed out into the lake. So of course if you do have more questions about stormwater issues, DEC does have a stormwater program. Maybe you're in contact with them, but they are certainly more knowledgeable about detailed stormwater issues than I am. And they have a lot of documentation on design help and things like that, which would be cool to check out. So yeah, I'll leave it there. I'm very interested in our discussion after this talk to hear kind of what type of things some of you might be working on in this arena. And then just to touch a little bit on kind of emerging topics related to cyanobacteria in Vermont that I think are interesting to keep an eye on. One of them is the issue of benthic cyanobacteria blooms. So the term benthic just means growing on the bottom. So these, rather than floating in the water, these benthic blooms are typically filamentous mat forming cyanobacteria that can sort of form these mats on lake bottoms, which might be released into the surface sometimes or might spend their whole life cycle attached to the bottom. So it's been found that benthic blooms it's safe to say they're getting more attention in the scientific community. It's not less clear whether they're actually expanding in their distribution, but it seems that they may be. There's just not a lot of data before the past few years on them. But what seems to happen is in a lot of cases where people do start to control nutrient inputs, so you might start to see some recovery from eutrophication. That causes the water to become clearer, but you still have a lot of nutrients stored in the sediments. And that's a good recipe for these bottom-dwelling cyanobacteria because they have enough light and they have access to all those nutrients stored in the sediments. So we're starting to see some benthic blooms in parts of Lake Champlain and some other Vermont lakes. We're also interested in understanding trends and patterns. Now that we're starting to get more data from Vermont lakes, we really want to start to understand things like which lakes bloom when, and ultimately why, with a goal of hopefully being able to predict and forecast blooms a little bit better. We're working on sort of incorporating cyanobacteria blooms into some of our regulatory structures in the state, particularly into the water quality standards. And we're looking to get a better understanding of the distribution of cyanotoxins in the state. So coming back to the issue of benthic cyanobacteria blooms, this is a photo from Lake Morey in Fairleigh, Vermont. So you can see these bands here. So these are something called Lingbia, which is a filamentous cyanobacteria you can see on the right there in a microscope image. So this typically grows attached to the sediment, but then later in the season, particularly as light starts to become limiting, some of those cells start to die. This causes gas bubbles to accumulate as they decompose, and this causes these mats to float up to the surface and form these big blooms. So this was a shock in Lake Morey. They hadn't seen anything like this for a long time. There had been blooms in the past there, but there had been an alum treatment, which was quite successful in reducing those blooms for a few decades. But over the past couple of years, they started to reemerge. In Lake Champlain, there was a number of observations a couple of years ago of this benthic colonies of this cyanobacterium called Calothrix, or it may be Rivularia. It's a bit hard to tell them apart microscopically. But this forms these kind of jelly-like globules that attach to rocks, and there were a number of them persisting for quite a while a couple of years ago. In 2022, this was a year in which the lake level was quite low, and I think that those low lake levels, may have increased light to the bottom in many areas and encourage these types of benthic blooms. And this year we also saw a lot of big aquatic plant nuisance issues in Lake Champlain, again, I think related to this low water level issue. So we're working on developing visual monitoring protocols to monitor for these benthic blooms. We need to start to develop criteria for severity, and we need to try to assess potential toxicity. Are these benthic blooms releasing toxins into the surface water? If so, how do we monitor for that, and when do they pose a threat to people? With respect to understanding trends and patterns, here I'm just looking at differences again in the different areas of the main lake, and what you're seeing is just the number of blooms reported on each day of the year. So for reference, August 1st would be day of the year 214 in these figures. What you can see here is some interesting things, like in the central main lake. This would be the part of the lake offshore of Burlington, for example. You typically have a lot of blooms going on around in July. So the middle couple weeks of July is when you have sort of peak bloom activity in that main lake section. You contrast that to the Northeast Arm, the Inland Sea section, or Missisquoi Bay. You have a much later peak of bloom incidence happening really in late August into early September. So there's a real difference in timing in different parts of the lake. And by analogy, this is also true for contrast between Lake Champlain and other lakes in the state, where some typically bloom sort of more in September into October. Others are kind of early summer bloomers. So I think a goal, is to start to develop a conceptual model of which lakes in the state are falling into which of those categories. And that can help us get a clue in terms of what factors might cause one lake or another to have a bad bloom year, so that we can help to manage that and predict that a little bit better. So another way to kind of look at trends is to look at spatial patterns and trends. So what you're looking at here is just the trends in those visual reports of blooms in different parts of Lake Champlain. So on the left panel here, you're looking at just kind of how bad blooms are. So this is really the spatial distribution of blooms over the monitoring period. And as you'd expect, you see the most yellow and red up in Missisquoi Bay and in Saint Albans Bay. In the right panel, you're looking at trends over time. So anything with an orange circle indicates that there seems to be more blooms happening over time. Anything with a blue circle indicates that there may be fewer blooms happening. So a couple things that jump out from looking at that is we certainly seem to see an increase in blooms in the sort of Burlington, Colchester area. And I think this lines up with some of the data we see on beach closures in Burlington and adjacent areas. In some of the areas like Missisquoi Bay and St. Albans Bay, we may may have some improvement, but the data here is really very noisy, so it's hard to say anything conclusive. Another exciting area, I think, for cyanobacteria monitoring is to increase our use of satellite technologies. So there are a couple products that the EPA puts out. This is one where you can get time series of cyanobacteria conditions from satellite images. This is a way we can start to gather data which are unbiased from a lot of lakes within the state. One problem we always have in interpreting our visual observation data is, of course, people are more likely to report when they see a bloom than when they don't see a bloom. So we never know kind of what's the real frequency of a bloom. Whereas if we're using a satellite imagery, of course, this is taking pictures whether or not there's a bloom there. So we can use that data to get a more unbiased picture of bloom frequency. Satellite data have had really limited utility up until recently because a lot of the pixel sizes were quite large, sort of 300 meters on a side, which meant that it was really hard to resolve things like shoreline blooms, which are the most common types of blooms we see in Champlain. But some of the new satellites coming online have resolution down to 0.5 meter, and those will really start to be able to see, sort of much smaller scale blooms and blooms in smaller lakes, where again, those larger, coarser resolution satellites haven't really been able to be very useful to us. So definitely interested in figuring out how we can use this data to kind of improve our monitoring, how to maybe flag lakes for increased monitoring, and again, get an unbiased picture of trends in the region. And also, satellite flyers have become a lot more frequent. So whereas we used to be able to get maybe weekly data, now many satellites are being offered at daily resolution at this high frequency. So that's pretty great. There's also a lot of exciting work going on in the basin in terms of forecasting cyanobacteria blooms. This is an emerging science. There aren't a lot of great bloom forecasting products out there, but there are a few. One of the kind of gold standards is the Lake Erie bloom forecasting system. There's been a lot of work, the number of institutions around Lake Erie, including NOAA, which generates this forecast product. And there's some interest at the level of NOAA and other institutions in developing forecasting products here. There's also a big effort underway at UVM, again, sponsored by NOAA, that I'm collaborating on trying to develop forecasts of blooms for Lake Champlain. So hopefully that'll work, and we're using a combination of approaches there. Here's this project is called the Cairo Project. That stands for Cooperative Institutes on Research on Hydrology. So this is a cooperation between several institutions, UVM being one, as well as university in some other states. There are a number of parts to this project. This includes forecasting of stream flow and flood events, as well as forecasting of harmful algal blooms, which is the part that I'm participating in. So this is quite a complex forecasting project where they're working on both process-based models where you try to simulate the movements of water, the differential equations that govern phytoplankton growth on the basis of nutrient limitation and light limitation, as well as sort of artificial intelligence-based approaches, which are sort of data-driven, heavy AI type models where you're trying to predict bloom occurrence on the basis of satellite imagery and other available data. So, and then there's hybrid approaches which kind of combine aspects of both of these. So we'll see what works the best, but hopefully something will come out that will provide kind of useful, actionable forecasts for the state. And the last thing I'll touch on is just our effort to expand cyanotoxin testing in the state. There are a number of cyanobacterial toxins. Of course, this is why we're concerned about cyanobacteria. These include things like microcystins, which is a common liver toxin, then cylindrospermopsins and anatoxins, which are both. Our cylindrospermopsin is also a liver toxin. Anatoxin is a neurotoxin, and all these things can cause acute sickness and even death in the right concentrations. Luckily, there have been no human deaths reported in in Lake Champlain, and those are pretty rare worldwide. But there are a number of cases where people will report gastrointestinal difficulties, breathing difficulties, and these can be from these or a range of other compounds that cyanobacteria produce. So GEC is taking over from the Vermont Department of Health for this cyanotoxin testing, and we have some increased funding to do this over the next few years. So we're currently monitoring for anatoxins and microcystins. We're going to add cylindrospermopsins and saxitoxins, which is another neurotoxin. And we're going to try to take a lot more samples from a lot more places to really get a sense of the distribution of these toxins in Vermont, which hopefully can help to inform the structure of our routine monitoring program going forward. So I don't know how I'm doing for time, but that's what I have for you. So I'd be happy to take any questions you might have.
Perfect. Thank you so much, Peter. We have a couple of questions. Our first one is from the Q&A, and Kate Kinney asks, the bloom that started in July this year in St. Albans Bay never completely went away. Does that mean the bloom laying on the shore will feed a new bloom in the coming spring?
So, I mean, this is a good question, and I have noticed that in St. Albans Bay is that in the last few years, it does seem like those blooms really suck in and never really let up. In St. Albans Bay specifically. So as far as whether that's gonna, I mean, I think the material that ends up on the shoreline is probably not going to be what seeds the bloom, but of course there's also a lot of material being left in the nearshore areas, deposited on the ground. I think particularly those cells that are resting in relatively shallow areas, but beneath the ice line are probably where a lot of those seed colonies are going to come for future years. So yes, this is it's a really interesting research topic in cyanobacteria is how they overwinter and how those overwintering colonies are recruited in subsequent years. Yeah, and certainly, all of the common cyanobacteria do have some strategies for overwintering. Dolichospermum and Ephanizomnin produce specialized resting cells called aconites, and you can see them a lot in the samples late in the season. And so these are essentially very durable resting stages meant to survive winter. I mean, they can survive in sediments for years and still reemerge. Microcystis doesn't produce these specialized cells, but they are also very much known to overwinter and have a whole stage of their life cycle resting on the sediment surface, which is not very well understood. It's a hard thing to study. So certainly there will be some recruitment of those colonies. I don't think anybody really has a great idea sort of to what extent a bad bloom year one year is going to result in a bad bloom year in the next year. I think it's not a perfect relationship. So I don't know if the number of resting colonies is really going to be the major determinant, but it's certainly going to play a role.
Okay, our next question is from Kent Henderson, and he asks, what measures dictate beach closures? Is it simply guided by visual parameters, or is toxin level testing needed to trigger a full beach closure?
So there's not really one answer to this. So because the authority to close beaches is really delegated to local health authorities. The state mostly does, advises local health officers in each town. So those are the authorities who really control closures of different swimming areas, or if it's a well-attended beach with beach managers, and they'll have different processes in place. But generally speaking, beaches are closed by visual parameters. It takes some time to get these toxin results back, and you don't want to wait for that and let people swim. So the question is really more whether beaches are reopened on the basis of toxins or visual confirmations. So in that sense, there are some beaches on places like Lake Carmi State Park where they typically wait for a negative toxin test to reopen a beach. Typically, those beaches use rapid tests, which are sort of test strips. I don't know if anybody remembers doing pH tests in science class where you use kind of a strip of pH paper. It's a similar type of test for cyanotoxins. They're not very exact, but they give a rough idea. So some localities use those, require a negative test strip before reopening. But generally speaking, I think our health department is comfortable with reopening a beach when there's no visual evidence of a bloom presence anymore. I mean, it's probably in an ideal world, you'd wait a day or so after the visual evidence of a bloom is dispersed, just to allow any residual toxins to wash away. But practically speaking, we have not detected high levels of toxins when you cannot see an active bloom going on. Yeah, that's a long answer to a short question. Sorry.
Okay, our next question is from Beth Daimling. Sorry if I'm mispronouncing your last name there. Do the toxins produced by cyanobacteria blooms disperse when the bloom disperses, or do they persist?
Yeah, I mean, I hope I maybe touched on a little bit of that in my response to the last question. I think there have been certainly reports in the literature of high toxins concentrated. They're being measured at relatively low cell densities. So when you don't see a lot of visual evidence of a bloom. In our monitoring data in Vermont, we haven't really seen that. But it's certainly possible. So that's why I think waiting a bit of time after a bloom disperses is probably a good idea. But generally speaking, fortunately, in Lake Champlain, we see pretty low concentration of cyanotoxins. We very rarely see concentrations above the health department's recreational guidance, which is good for the cyanotoxins that we've tested for. So maybe once or twice a season, we'll get a reading like that. Many seasons, we don't get any readings like that. So it's certainly possible that there's some residual cyanotoxin, but probably not enough to be dangerous from a recreational perspective. From a drinking water perspective, it could be a different issue that would be more sensitive. But drinking water systems are pretty good at neutralizing cyanobacteria toxins, unless they get to pretty high concentration. So that has not been an issue in our drinking water testing in the state.
Okay, we have another question from Dean Pierce. He asked, our approach to managing cyanobacteria seems to be aimed at, if you can pardon the phrase, starving them by reducing nutrients. Do they have any vulnerabilities that might provide other avenues for management?
No, I mean, it's tricky. And cyanobacteria blooms are a tricky problem. And, there's a reason that most places in the world focus on phosphorus controls for this, is because it really seems to be the most effective way to do it. There are other strategies that are attempted. Mostly it still comes down to reducing phosphorus, but in some situations, a lot of that phosphorus is being released from the lake sediments. So in places like Missisquoi Bay, where I've done a lot of research, you can really see a strong signal of internal nutrient loading. So when you get the right conditions, you get oxygen consumed in those sediments. This causes dissolution of iron compounds. Essentially, they dissolve. And so all the phosphorus that's sticking to the surface of those particles is then released into the water column. And this is sort of the process we call internal loading. This occurs in, all this phosphorus originally came from the catchment. So the dichotomy between internal versus external sources is a bit misleading. It's all external phosphorus on the right time scale, but when we're talking about what's happening at the scale of a given season or even a given decade, that's when this distinction becomes important. So there are approaches to try to reduce that internal load. This is, I alluded to an alum treatment in Lake Maury in my talk. So this was a case where they poured alum, which is an aluminum compound that binds phosphorus irreversibly. So whereas that iron is capable of dissolving when in the absence of oxygen, once phosphorus sticks to these aluminum compounds, it's pretty much permanently bound, unless you get really high or really low pH. So this is a common lake management practice, is to put either alum or lanthanum based product, which is similar, to try to bind that phosphorus and prevent its release from sediments. So this is more of like a direct intervention than trying to slowly reduce landscape sources. But this is also really only practical if you're also reducing landscape sources at the same time, because otherwise you're just going to bury that alum with new reactive phosphorus sediments, and the problem will start all over, and you'll end up paying a lot of money for reducing blooms for 2 or 3 years. So that's really not something that anybody wants to invest in, and it's not a great strategy. So yeah, I mean, starving them is really the way to go. We might be able to improve our efforts in this respect by getting a better understanding of which parts of the phosphorus load we're targeting when we target our nutrient load reductions. So just like you could put carbon into a system in the form of coal, or you could put carbon into the system in the form of bread or marshmallows, and those things are going to be differently available to the things that would eat them, so similarly with phosphorus, there's some phosphorus that's coming in and is not very available. It's bound to sediments in a pretty irreversible way. It's probably going to settle out for the most part and not really be a big problem. Whereas if you have really reactive dissolved phosphorus compounds, those are definitely going to be available to plankton when they get to the lake. So targeting nutrient loads, associated with those really reactive phosphorus fractions is, might be more effective and more bang for your buck than some of the kind of broader total phosphorus nutrient load measures. So that's certainly something that's under discussion.
All right, we've got another question from Kate. She asks, are we able to monitor the effect of recreational boating on the development of cyanobacteria?
Yeah, well, monitoring it would be tricky. That would require a pretty sophisticated setup to measure sort of in real time the turbulence associated with boats. I think, perhaps some of you have heard of the current issues with trying to develop regulations for wake boats in the state. There have been some studies with wake boats which generate a really large wake and that propagates deep into the water. That may increase nutrient loading from the sediments by just either disturbing the sediments or just increasing the sort of pressure oscillations of the sediment water interface, which can pump out some of that pore water. And it can resuspend sediments, particularly in the shallow areas near the shoreline, which can, indirectly reduce cyanobacteria or increase cyanobacteria by reducing other aquatic plants. If you dislodge the things that are competing for those nutrients, you might allow more nutrients in the water for the cyanobacteria to take up. But getting good data on that, that would be really tricky to do and probably beyond our capacity at DEC to do a good job of it. That's probably something that would have to be funding to contract out to a university or somebody else interested in trying to get a handle on it. And yeah, there have been reports of things like wake boating increasing, phosphorus concentrations in some lakes where there's really heavy wake boat use, particularly shallow lakes, in other parts of the U.S., but not probably on the type of boating activity of the scale that we typically see here in Vermont.
I have a question. You mentioned that a lot of nutrients are coming from urban areas. I was wondering, are there certain products we're using in our households, like laundry detergents or car wash, stuff like that, that are contributing to increase nutrients?
I think laundry detergents, you see phosphate free on many of them. And I think that was a major target of early Clean Water Act regulation, was the reduce of these phosphate-based detergents, because that was a major source of phosphorus pollution in kind of the 70s, 60s period. And that caused a lot of the sort of big water quality problems that prompted the implementation of the Clean Water Act, excuse me. In terms of common household things now, I mean, certainly the obvious one is any kind of lawn fertilizer. Those lawns are not good environments for infiltration. So if you have lawn fertilizer and it rains, a lot of that water is going to carry it right off. Um, that's not, so that's one thing that will have a direct impact. And of course, those are pretty available nutrients. So just as they're available to the grass, they're going to be pretty available to the cyanobacteria once they get to the water bodies. In terms of other compounds, yeah, I mean, there may be, it's a good question, but yeah, in terms of household cleaning stuff, I think, not so much anymore. But someone will probably at some point prove me wrong on that. I don't know.
All right, so you have two more questions in the chat. The first one is from Jeff, and he asks, do you have any information about the specific role of septic tanks?
Yeah, I mean, so the role of septic tanks, there have been some studies of that. It's been brought up a lot. And the studies that have been done in Vermont have suggested that they're probably, in most places, not a major contributor. And this is because most of the sediments in Vermont just have a lot of capacity to bind phosphorus. So once that phosphorus is down in the water, even if it's leaching out of the septic tank into the surrounding sediments, that sediment is going to hold on to that phosphorus really well, and it's not really easy to mobilize that. Now, there might be cases, where you get flow of some of that septic, leachate through what they call macropore flow, or larger channels within the sediment matrix, where it won't interact as much with those sediment particles. So sort of think of it as a crack in the sediment. This is sometimes associated in the hydrological literature with sort of freezing and thawing, and you might, after a lot of freeze and thaw cycles, you see these cracks in the soils, and water gets into those. It can flow out really quickly and not interact with the sediment. This might happen in some places deeper in the sediment profile, in which case you might get some leaching from septic systems. But again, there's not a lot of evidence for it. My colleague who runs our volunteer-based lake chemistry monitoring program, the lake monitoring program, whose name is Mark Mitchell, tried to do a pilot study looking at caffeine concentrations in a number of lakes in Vermont. And caffeine can be used as a proxy of that septic input because it breaks down relatively quickly, but it doesn't bind to the sediment. So it will get flushed out into the lake. So if you can see a lot of caffeine in the lake, that's a pretty good indication that there's wastewater getting into it. I know in the lakes that he tested, he did get a couple of positive hits in a couple of places. There might be an issue with the detection limits, but it's also hard to correlate that with the exact amount of phosphorus coming into the lake. So it's a cool proof of concept, and it's suggestive that there could be a problem. But in terms of understanding the scale of the problem, it's a tricky thing to do. Any of this kind of groundwater input work is hard to carry out well. I would say in some areas that have, sandier soils, you might have a larger input of this septic system phosphorus. In areas where you have a lot of clay in your soil, you're probably less likely to have it. So it really depends on that soil composition.
So Kent had another question from the chat, and that was a decade ago, a hypothesis was presented by some environmental groups that there was a connection between ALS and harmful algal blooms. Has any new supportive evidence emerged to prove or disprove that hypothesis?
Yeah, I mean, this is a bit of a contentious issue for some. I mean, not for me. So there's a compound called BMAA, which is essentially a modified amino acid produced by many cyanobacteria, which has been implicated in ALS. This has been looked at, and there are some very strong adherents to this theory who think, that it's a major cause. but there have been some reviews which really have cut this down a lot, basically citing both methodological problems with some of the studies advocating this hypothesis and sort of issues with the epidemiological kind of methods, I guess. So both the analytical and the epidemiological methods, might be somewhat questionable in some of the research supporting this hypothesis. Basically, there does seem to be some evidence, although again, I don't know if this is 100%, that at high enough doses, this BMAA does seem to accumulate in the brains of ALS or neurodegenerative disease patients with ALS-like symptoms. And there have been some studies suggesting that there are clusters of these cases around some lakes that have cyanobacteria blooms. But again, those studies are really quite problematic or maybe problematic because there are many, many lakes with cyanobacteria blooms, and certainly not many or most do not have these types of clusters around them. And so proving, you'd have to have a really robust data set to understand kind of whether these clusters are more than you would expect by chance, because just random distribution of these type of diseases are not, might not appear random at first. They tend to cluster just naturally. Again, this is based on my reading of some review papers that the EPA and other people have put out, because we do get a number of questions about this issue. I think the basic opinion that I've come away from, and with the caveat that I'm not a toxicologist, and so I try to read the literature and understand the consensus, but I think that it's kind of too early to say. I think I tend to be skeptical of the literature that talks about kind of airborne cyanotoxins in this, relative to this BMAA hypothesis, because I think the amount being aerosolized is going to be so low once you get even a few meters away from the lake that probably wouldn't be a major health concern. But yeah, I mean, so I'm sure there has been more supporting evidence as well as more disproving evidence presented this active research area. And, I think it's always ongoing, but it's certainly not a settled topic yet.
Any more questions floating around out there? All right, looks like we're a little past 7 o'clock. If there's no more questions, I think we're all good. But once again, I want to thank you so much for being here, Peter. That was a wonderful presentation. Thank you to everybody who attended. And if you'd like access to a recording of this presentation, as well as more information on Franklin County Stormwater, there's a link in the chat. So once again, thank you all so much.
Yes, thank you, Peter, and thank you everyone who came and asked questions.
Yeah, you're very welcome, and spread the word about becoming monitors if anybody's interested.
Yes, absolutely.